Welcome back, everybody, for another deep dive. And this time, we are going to get into like a real-world detective story.
Oh, cool.
Yeah, it's the journey of a successful HIV protease inhibitor drug.
Okay.
And we are going to go through like all the stages of drug discovery. So from, like identifying the target
Right.
to actually like optimizing the final product.
Yeah.
And, to make things even more interesting, we're going to compare this success story
Okay.
to a drug that, didn't quite make it.
Oh, interesting.
Yeah, it's a great way to see like what separates the winners from the
From the losers.
yeah, let's say the not so winners in this world of high stakes.
Right. High stakes research.
Exactly. So let's start with the basics.
Okay.
What is HIV protease?
Okay.
And why was it such a promising target for researchers, battling HIV?
HIV protease is an enzyme, and it's essential for the virus to replicate.
Okay.
So without it, new virus particles can't mature and spread. So think of it kind of like a crucial worker on the HIV production line.
Okay.
You knock out that worker, you halt the entire factory.
So disrupting HIV protease could like effectively stop the virus in its tracks.
Yes.
Did research back this up?
Yeah, absolutely. Research published in the late 1980s, particularly studies like the one by Kramer et al.
Okay.
In 1989, showed that inhibiting HIV protease could block the virus in lab settings. So this was, a major breakthrough
Yeah.
and a huge validation of HIV protease as a target for drug development.
Okay, so target validated.
Yes.
I imagine that sparked a lot of excitement in the research community.
Oh, yeah.
But then comes the real challenge. How do you actually design a drug
Right.
to do that, to block HIV protease?
Right. So the initial design phase involved understanding the structure of HIV protease.
Okay.
And its mechanism of action. So researchers relied heavily on techniques like X-ray crystallography and computer modeling to visualize the enzyme's active site,
Okay.
the region where it binds to its substrate.
It's like trying to design a key for a lock you've never seen before,
Exactly.
but with incredibly high stakes.
Yeah, you need a key that fits perfectly,
Right.
blocks the lock, but doesn't accidentally open any other locks in the process.
Right, of course, yeah.
Scientists needed a molecule that bound specifically to HIV protease
Right.
and interfered with its function without affecting other important enzymes in the body.
So they had to find a molecule that was both potent and selective.
Yes.
What were some of the early approaches they used to tackle this?
So one of the earliest approaches involved mimicking the natural substrate of HIV protease,
Okay.
the molecule it normally interacts with.
Uh-huh.
So the idea was to design a molecule that resembled the substrate closely enough to bind to the enzyme,
Okay.
but different enough to prevent it from functioning properly.
So it's like a decoy,
Yeah.
tricking the enzyme into binding to it
Yes.
instead of its intended target.
Exactly.
Wow.
So researchers designed peptidomimetic inhibitors, molecules that mimicked the peptides normally cleaved by HIV protease.
Got it.
And these early inhibitors, like the one described by Roberts et al. In 1990, showed some promise, but they faced challenges with stability and bioavailability.
Bioavailability, that means how well the drug is absorbed and reaches its target in the body, right?
That's right. It's not enough for a drug to be effective in a test tube. It needs to reach its destination in the body at sufficient concentrations.
Right.
Yeah.
So they had a promising starting point, but there were definitely hurdles to overcome.
Yeah.
It's like having a rough sketch of the key, but needing to refine it, make it more robust and durable.
Yeah.
This is where lead optimization comes in, right?
Exactly. This stage is all about fine-tuning the drug molecule,
Okay.
tweaking its structure to enhance its potency, selectivity, and bioavailability. It involves a lot of trial and error testing, different modifications, and analyzing their effects.
So let's imagine they have a few promising lead compounds. How do they go about optimizing them? Sounds like a delicate balancing act.
It is. Think of it like adjusting a recipe.
Okay.
You might add a pinch of this, a dash of that.
Right, yeah.
Constantly tasting and refining until you achieve the perfect balance of flavors.
Yeah.
In drug development, researchers make subtle changes to the chemical structure of the lead compound,
Okay.
adding or removing functional groups, tweaking its shape, or even modifying its overall size.
And each modification is like a tiny experiment, seeing how it affects the drug's behavior, right?
Yes.
Testing how well it binds to the enzyme, how stable it is in the body, how it's metabolized, and so on.
It's a constant process of testing, analyzing, and refining, driven by data and guided by a deep understanding of chemistry and biology.
Yeah.
And as we explore the specific modifications made to this successful HIV protease inhibitor, you'll see just how intricate this process can be.
This is where the real detective work comes in.
Yeah.
You have to follow the clues, rule out the dead ends, and eventually hopefully crack the case.
Exactly. And to add another layer of intrigue to our story, we'll also be looking at a drug that went through a similar process but didn't quite make it to the finish line.
That's what I'm really curious about.
Yeah.
What makes one drug successful while another seemingly similar one falls short?
Yeah.
I'm ready to hear all about it.
Let's dive into the specifics of our successful drug.
Okay.
It went through this rigorous lead optimization.
Right.
And one of the key challenges was improving its bioavailability.
Okay.
Early versions of the drug weren't absorbed well in the digestive system, so this limited their effectiveness.
So how did they tackle that? Did they change the drug's formulation or try, like, a different delivery method?
They explored several avenues.
Oh.
One strategy involved modifying the drug's chemical structure
Okay.
to increase its solubility and permeability.
Okay.
For instance, they introduced lipophilic groups,
Okay.
which essentially made the drug molecule friendlier to fats, allowing it to pass through cell membranes more easily.
It's like giving the drug molecule a special passport
Yeah.
to cross borders more efficiently.
Exactly.
I like that.
These modifications, as described in Kempf et al. In 1995,
Okay.
significantly boosted the drug's oral bioavailability, making it much more effective when taken as a pill.
That's a huge win.
It is.
But bioavailability is just one piece of the puzzle.
Right.
What about other aspects like potency and selectivity?
Yeah.
Were there any modifications made to enhance those?
Absolutely. Researchers found that by adding certain chemical groups to the drug molecule,
Okay.
they could enhance its binding affinity to HIV protease, making it a more potent inhibitor.
Okay.
For example, incorporating a hydroxyethylene moiety,
Okay.
as reported by Vaca et al. In 1994,
Okay.
significantly increased the drug's ability to block the enzyme.
So they were essentially fine-tuning the key to fit the lock even more snugly,
Yeah.
preventing the enzyme from doing its job.
Exactly.
But what about selectivity? How did they ensure that the drug targeted HIV protease specifically and didn't interfere with other enzymes in the body?
Selectivity is crucial.
Yeah.
To minimize side effects,
Of course.
researchers carefully analyzed the drug's interactions with other enzymes, looking for any off-target binding.
Yeah.
They made strategic modifications to minimize these interactions. For instance, they found that by replacing a certain bulky group with a smaller one,
Okay.
they could reduce the drug's affinity for a related enzyme,
Got it.
improving its selectivity.
It's like fine-tuning a musical instrument,
Yeah,
making sure each note is clear and distinct with no unwanted dissonance.
that's a great analogy.
Thanks.
Through these meticulous modifications, they sculpted the drug molecule into a highly potent, selective, and bioavailable weapon against HIV protease.
Wow.
But now let's turn our attention to the failed drug.
Yeah.
It's the one that didn't quite make the cut.
The cautionary tale. This drug also targeted HIV protease, right?
Yes, it did.
What went wrong in its development? Did it stumble at a particular stage, or was it a combination of factors?
This drug went through a similar design process.
Okay.
And early studies showed promise. It exhibited good binding affinity to HIV protease,
Okay.
and even demonstrated some antiviral activity in lab tests.
So it seemed like they were on the right track. What happened?
The problem emerged during clinical trials.
Oh.
It turned out that this drug had a major drawback.
Okay.
It was rapidly metabolized in the liver.
The body's detoxification center.
Right.
It's like having a highly trained cleanup crew that's a bit too efficient.
Yes.
Removing the drug before it can do its job.
Exactly. The drug was broken down so quickly that it couldn't reach therapeutic concentrations in the bloodstream.
Wow.
So despite its initial promise, its rapid metabolism rendered it ineffective in patients.
So it's a classic case of a drug that looked good on paper,
Yeah.
but failed the real-world test.
Precisely. This highlights the importance of understanding a drug's pharmacokinetic properties.
Okay.
How it's absorbed, distributed, metabolized, and excreted. Even if a drug binds perfectly to its target, if it can't reach that target in the body at sufficient levels, it's destined to fail.
It's a crucial reminder that drug discovery is not just about finding a molecule that hits the target.
All right.
It's about understanding the entire journey that drug takes within the body.
Absolutely. And as we've seen comparing these two drugs, one a resounding success,
Right.
and the other a disappointing failure, helps us understand the critical factors that separate triumph from defeat in the world of drug development.
So our Goldilocks drug, with its optimized potency, selectivity and bioavailability emerged victorious.
It did.
What about its impact?
Yeah.
How did it change the landscape of HIV treatment?
So this drug really did arrive kind of like a knight in shining armor.
It did.
You know,
Yeah.
it was a game changer.
Yeah.
It marked a turning point in the fight against HIV/AIDS.
It did.
It wasn't a cure,
Right.
but it offered something incredibly valuable.
Yes.
Hope, exactly.
So, tell me a little bit more about that.
So it was one of the first HIV protease inhibitors to demonstrate this remarkable efficacy in clinical trials.
Okay.
And the results were striking patients taking this drug experienced a significant reduction in viral load, a measure of the amount of HIV in the blood.
Okay.
And in many cases, the virus became undetectable.
Wow. So it's like pushing the virus into hiding, giving the immune system a chance to recover.
Exactly. And this translated into tangible benefits for patients.
Okay.
They were less likely to develop opportunistic infections, those infections that take advantage of a weakened immune system.
Right.
So their overall health improved and their life expectancy increased dramatically.
So it wasn't just about adding years to life. It was about adding life to years.
Exactly.
Transforming HIV/AIDS from a death sentence to a manageable chronic condition.
Precisely.
This drug, along with other protease inhibitors that followed, truly did revolutionize HIV treatment.
It did.
It ushered in the era of HAART,
Yes.
highly active antiretroviral therapy, a combination therapy that targets different stages of the HIV life cycle.
That's right. It's like a multi-pronged attack, hitting the virus from multiple angles, making it much harder for it to develop resistance.
And this approach has been incredibly successful.
It has.
HAART has significantly reduced the incidence of AIDS-related deaths
Yes.
and has dramatically improved the quality of life for millions of people living with HIV worldwide.
Absolutely.
The testament to the power of scientific research.
It is.
And a reminder that even in the face of seemingly insurmountable challenges, breakthroughs can happen.
Yes.
But as we've seen with the failed drug,
Right.
the journey is not always smooth.
Right.
What are some of the ongoing challenges in HIV drug development?
So one of the biggest hurdles remains drug resistance.
Okay.
HIV is a highly mutable virus, constantly changing and adapting. It can develop mutations that make it less susceptible to the drugs we use against it.
So it's like this constant arms race.
Yes.
Trying to stay ahead of the virus as it evolves.
That's a great way to put it. Researchers are constantly working to develop new drugs that can overcome resistance.
Okay.
Exploring novel targets within the virus and designing drugs with different mechanisms of action.
So it's a continuous process of innovation driven by the need to stay one step ahead of this formidable foe.
It is.
What about the future? What's on the horizon for HIV treatment and prevention?
The research landscape is incredibly exciting. Scientists are working on long-acting injectable formulations of antiretroviral drugs, which could reduce the burden of daily pill taking.
Yeah.
There's also a lot of research focused on gene editing techniques, exploring the possibility of actually removing HIV from infected cells.
That sounds like science fiction becoming reality.
It does, doesn't it?
And what about preventative measures? Are there any new developments in that area?
Pre-exposure prophylaxis, or pre-AFP, has been a major breakthrough in HIV prevention.
Okay.
It involves taking antiretroviral drugs daily to reduce the risk of contracting HIV. Newer pre-RP medications are even more effective and have fewer side effects.
There's also ongoing research into developing a vaccine for HIV, which would be a game changer in the fight against the virus. So while there are still challenges, there's also immense hope for the future.
Absolutely.
The progress we've made in HIV treatment and prevention is a testament to the power of scientific ingenuity, collaboration, and perseverance.
I couldn't agree more. And it's a reminder that scientific research can truly make a difference in people's lives.
Yeah.
It's a story of triumph over adversity, a story that continues to unfold as we strive to create a world free from HIV/AIDS.
We've covered a lot of ground today.
We have.
From the intricate molecular details of HIV protease to the transformative impact of a successful drug and the ongoing challenges that lie ahead.
Yes.
What's the one key takeaway you'd like our listener to ponder as they go about their day?
I'd say the story of HIV drug development is a powerful example of how scientific curiosity, rigorous research, and a deep understanding of both chemistry and biology can lead to life-saving breakthroughs. It's a story that highlights the importance of continued investment in scientific research.
Yes.
And the potential for science to change the world for the better.
Beautifully said. It's a story that inspires hope and underscores the power of human ingenuity. Thanks for joining us on this fascinating deep dive.
It was my pleasure.
Until next time, stay curious and keep exploring.